Good afternoon, everyone. So I will be speaking regarding the primary data from Destiny Lung01 study. So as we all know that HER2 mutations drive around 3% of non-squamous non-small cell lung carcinoma and are associated with slightly younger age, female sex and never smoking history, and they are associated with a poor prognosis and an increased incidence of brain metastasis. So before this trial, there were no approved HER2 targeted therapies for patients with non-small cell lung carcinoma. And HER2 metastasis status is not routinely checked, and this population is treated with standard chemotherapy and immunotherapy. So the efficacy in a second and third line setting is quite limited, with the overall response rate in the range of 7% to 27%. And Trastuzumab deruxtecan is a HER2 antibody drug conjugate approved in various countries worldwide for the treatment of metastatic HER2 positive breast cancer and gastric cancer. So this Destiny Lung01 trial, they included the patients who were unresectable metastasis, non-squamous, non-small cell lung carcinoma, and they had a EOG status of 0 and 1, and it was a multi-center international phase 2 cohort study which included HER2 overexpression as well as HER2 mutation. So the baseline characteristics were at the time of analysis they had recruited total 91 patients, and the median age was 60, with the youngest patient being 29 and the elder one was 88. They included 65.9% of female patients, and ECOG performance status was 0 and 1, and HER2 mutation was kinase domain mutation was noted in 93% patient. 36.3% had an asymptomatic CNS metastasis at baseline. Around 57% of patients were never smokers, and around 22% of patients had a prior history of lung resection. So out of the 91 patients, 90 patients had history of prior systemic therapy, with the median number of prior lines were 2, ranging from 0 to 2. So these were heavily treated patients, which included platinum-based therapy, PD-L1 based therapy, docetaxel, and also around 14.3% of patients had HER2 TKI prior. So coming to the response assessment, so the primary objective was overall response rate, objective response rate. So it was around 54.9%. Other secondary other secondary endpoints were the PFS and OS, which I will discuss later, and the DCR, disease control rate, was around 92.3%. So this is the curve showing the amount of response. So we can see that responses were observed across all HER2 mutation subtypes as well as in patients with no detectable HER2 expression or HER2 gene amplification. So and the response of Trastuzumab deruxtecan was uniform across all subtypes. Those who received prior HER2 TKI or who received platinum-based therapy or PD-L1 therapy. It was also it has shown an acceptable outcome in patients with asymptomatic CNS metastasis, whether they have received prior RT or not. So this was the spider plot showing the responses. So coming to the PFS, the median PFS at the time of analysis was 8.2 months. The median OS was 17.8 months, and in the safety profile, there were incidents of serious drug-related adverse events in 19.8% patients, and in 25.3% patients, it lead to discontinuation of the drug due to drug-related adverse events. So these were the altogether the major adverse events. Commonly noted were nausea, fatigue, alopecia, vomiting, along with hematological toxicities, most commonly neutropenia or leukopenia. But the adverse events of special significance were ILD and pneumonitis. So any grade ILD or pneumonitis were seen in around 26.54% of patients, and the median time to the onset of first reported drug-related ILD or pneumonitis was 141 days, ranging between 14 days to 462 days, and the median duration of 43 days. So 75% of drug-related ILD or pneumonitis were low grade, especially it was grade 1 and or grade. 24 patients with drug-related ILD or pneumonitis received more than or equal to 1 doses of glucocorticoids. However, not all glucocorticoid treatment was administered per the ILD or pneumonitis management guidelines. So at the time of data cutoff, 54% of investigator reported cases had fully resolved. So they conclude that the Trastuzumab deruxtecan demonstrated a robust and durable anti-cancer activity in patients with previously treated HER2 non-small cell lung carcinoma. Efficacy was consistently observed across all subgroups, including those with stable CNS metastasis. Exploratory analysis demonstrated anti-cancer activity across different HER2 mutation subgroups as well as in patients with no detectable HER2 expression or HER2 gene amplification. Overall, the safety profile was consistent with previously reported studies. Most adjusted drug-related ILD or pneumonitis cases were of low grade, and ILD or pneumonitis remains an important identifiable risk, and effective early detection and management are critical in preventing. So because the original dose used was 6.4 milligram per kg, so they are evaluating lower dose, that is 5.4 milligram per kg dose in the future study of DESTINY-02. So DESTINY Lung01 provides compelling evidence of positive benefit and risk balance with Trastuzumab deruxtecan in the second line setting and supports its establishment as a potential new treatment standard. Thank you.